Transcription is the process where a section of DNA, known as a gene, is used to create an RNA molecule. This process all occurs in the nucleus, and one gene produces one RNA molecule, which is a copy of a gene. There's three separate steps in transcription, and the first step is referred to as initiation. In initiation, the two strands of DNA are unwound. So the double helix is actually unwound. The base pairs are broken apart so that the strand starts to come apart. And this exposes a template strand. Then an enzyme called RNA polymerase, represented by this large purple image here, binds to something which is called a promoter. Now, promoter is just the name for a region of DNA in which RNA recognizes a particular sequence of bases, which is referred to as a promoter region. The second step in transcription is called elongation, and that's where the RNA polymerase molecule actually moves from the 3' end to the 5' end of the DNA template strand. So this bottom strand here is the DNA template strand, which is then coding for the RNA. Now, as that RNA polymerase moves along the template strand, what it's doing is taking nucleotides, free nucleotides that are moving around in the cytoplasm, and it's joining them together to form the RNA molecule. Except, of course, that in RNA, thymine is replaced by uracil in the sequence. Now, when that RNA is being built, it is of course being built from the 5' end of the RNA sequence to the 3' end. So this would have been the very first nucleotide that was added to the sequence, and this at this end here, the 3' end, would have been the most recent nucleotide that's added to the sequence. The third and final step in transcription is called termination, and that's when the RNA polymerase reaches a section called the terminator sequence. And this sequence indicates the end of the gene so that the polymerase knows when to stop coding the RNA. The polymerase, once reaching that sequence, then moves off. It separates from the DNA strand and it releases the new RNA into the surrounding area of the nucleus. The DNA then winds itself up again into the double helix shape. The RNA becomes one of three different molecules depending on the shape it forms, which could be mRNA, tRNA, or rRNA, and that all depends on the three-dimensional shape that RNA folds into. mRNA is short for messenger RNA, and messenger RNA has the specific job of translating into proteins. So mRNA codes for proteins. In prokaryotes, ribosomes can actually begin translating mRNA into a protein even before transcription is complete. But in eukaryotes, the mRNA has to be altered in two specific ways before it can leave the nucleus and be translated in the cytoplasm. One of those processes is the removal of introns, and introns, represented by these green segments here, are portions of a gene that don't actually encode for an amino acid sequence, which means they're useless in coding for a protein. They're not needed and they have to be removed. The only sections of a gene that are useful for coding an amino acid sequence are exons. So what you want is to remove all of the introns in green so that you're left with just the exons in yellow. So of course when you transcribe DNA to RNA, those introns are also transcribed. So you end up with introns in your RNA. And RNA that contains both exons and introns is referred to as pre-mRNA. In order for pre-mRNA to become mRNA, those introns need to be spliced out. You can see in this final mRNA molecule, it's yellow, meaning that it only contains exons. The other important alteration before pre-mRNA becomes mRNA is the addition of a methylated cap or a methyl cap on the 5' end of the pre-mRNA and something referred to as a poly-A tail, which is added to the 3' end of the pre-mRNA. Together, the cap and poly-A tail enhance translation in the next step in the cytoplasm by helping ribosomes attach to the 5' end of the mRNA molecule. The length of the poly-A tail may also determine how long an mRNA lasts before being degraded. What you end up with is an mRNA molecule which has a 5' methylated cap, a poly-A tail at the 3' end of the mRNA. It only contains exons, which are sections that will be coding amino acids. It's removed the introns, and then it can move out into the cytoplasm for the next step in protein synthesis, which is translation.